Welcome to Unit 9, which is about developmental psychology. As we journey through life, when and how do we develop? Developmental psychology examines how people are continually developing, both physically, cognitively, and socially, from infancy through old age. Much of the developmental psychology's research centers on three major issues of nature-nurture, continuity in stages, and stability in change. Here's what you should be able to do by the end of the day, and we will quiz at the end of the Module 45 today. Identify three issues that have engaged developmental psychologists. I just gave you a heads up as to what they will be, and they'll be on the leading screen, I believe. Discuss the course of prenatal development and how teratogens affect development, and describe the abilities of newborns, noting how researchers are clearly able to identify mental abilities. So the first question asked you about psychology's three major issues, and I gave you an intro, but here we go. Number one, nature versus nurture. How does genetic inheritance, our nature, interact with our experiences, which is our nurture, to influence our development? Secondly, continuity in stages. So what part of development are gradual and continuous, like riding an escalator, and what parts does our body and our psychology kind of change abruptly, like climbing rungs on a ladder? And finally, stability and change. Which of our traits persist throughout our lifetimes, and how do we change as we get older? Nature and nurture. The gene combination created when our mother's egg engulfed our father's sperm helped form us as individuals. Genes predispose both our shared humanity and our individual differences. But is that also true for our experiences? But it's also true that our experiences form us. In the womb, in our families, and in our peer social groups, we learn ways of thinking and acting, and even differences initiated by our nature may be amplified by our nurture. So we're not formed by either nature or nurture, but by their interrelationships, their interaction with one another. Biological, psychological, and social cultural forces all interact together to create us. Mindful of others' differences, however, we often fail to notice the similarities that are stemming from our shared biology. Regardless of our culture, humans share the same life cycle. We speak to our infants in similar ways and respond similarly to their coos and their cries. All over the world, children of warm and supportive parents feel better about themselves and are less hostile than children of punishing and rejecting parents. And although ethnic groups differ in school achievement and delinquency, the differences are no more than skin deep, to the extent that family structure, peer influences, and parental education predict behavior in one of these ethnic groups, they do so for others as well. Compared with person-to-person differences within groups, the differences between groups are relatively small, and something for us to remember and think about. Continuity in stages. Generally speaking, researchers who emphasize experience and learning see development as a slow, continuous shaping process. Those who emphasize biological maturation tend to see development as a sequence of genetically predisposed stages or steps. Although progress through the various stages may be quick or slow, everyone passes through stages in the same order. So are there clear-cut stages of psychological development, as there are physical stages, such as walking before running? Now I want to introduce you to a couple of people who are famous for their theories around these ideas. And the first one is Lawrence Kohlberg. Lawrence Kohlberg has theories around moral development, and you can see at the very top his blue tab, pre-conventional morality, conventional morality, and post-conventional morality. And he suggests that there are people that never reached post-conventional morality. So he would say that the process is slow and gradual, and those stages are fairly concrete, and that some people never reach post-conventional morality. So we'll take a look at him in a little bit. Erik Erikson is really a famous guy in education because a lot of education is built around his stages, which we'll go through, and you might be assigned a project on one of his stages. So if you take any sort of educational psychology at all, if you go into education for any facet, you will be well versed in Erik Erikson. And then finally, Jean Piaget, who you've already met. Jean Piaget is all about cognitive development, and he has theories about like why babies behave the way that they do and why we are like we are in various stages of life. Some research casts doubt on the idea that life proceeds through these neatly defined age-linked stages. Young children have some abilities that Piaget attributed to later stages, and Kohlberg's work reflected a worldview characteristic of individualistic cultures and emphasized thinking over acting. And adult life does not progress through fixed, predictable series of steps all the time. Chance events happen and influence us in ways that we couldn't have predicted. Nevertheless, the concept of stage remains pretty useful. The human brain does experience growth spurts during childhood and puberty that correspond roughly with Piaget's stages, and stage theories contribute a developmental perspective on the whole lifespan by suggesting how people of one age think and act differently when they arrive at a later stage. Conception. Nothing's more natural than a species reproducing itself, and nothing is really more wondrous. With humans, the process starts when a woman's ovary releases a mature egg on the left. A cell roughly the size of a period at the end of this sentence. The woman was born with all the immature eggs that she would ever have, although only 1 in 5,000 will ever mature and be released. A man, in contrast, begins producing sperm cells at puberty, and for the rest of his life, 24 hours a day, he will be in a non-stop sperm factory with the rate of production, in the beginning, more than 1,000 sperm during the second it takes to read this phrase, slowing down with age. So women born with all the cells that they're ever going to have, and males producing sperm consistently throughout their lives. Well, fairly consistently. So like space voyagers approaching a huge planet, 200 million or more sperm begin the race upstream toward an egg, approaching the cell 85,000 times its own size. The relatively few that reach the egg release digestive enzymes that eat away its protective coating, and as soon as one sperm penetrates that coating and is welcomed in, the egg surface blocks out the others. Before half a day elapses, the egg nucleus and the sperm nucleus have fused. The two have become one. Consider it your most fortunate moments. Among 200 million sperm, the one needed to make you, in combination with that particular egg, won the race. And so it was for innumerable generations before us. If any of our ancestors had been conceived with a different sperm or egg, or died before conceiving, or not chanced to admit the partner, it would have broken a chain of events that produced us.
They're full of the same tired tropes. For example, when sperm meets an egg, there's a literal spark. I'm so over it. Hello everyone, I'm Crystal here for D-News. Recently, researchers from Northwestern University reported the discovery of a zinc spark in human oocytes. This phenomenon, known to occur in other mammalian species, is the result of sudden releases of zinc after egg fertilization. Before mammalian eggs or oocytes are either released by the ovaries or harvested by a doctor, they absorb billions of zinc atoms that act as a signal for them to halt meiosis cell division and wait to be fertilized. They're in a holding pattern until they meet a sperm, and if they do meet one, at that moment of fertilization, several things have to happen all at once. First, the egg has to protect itself from the insertion of DNA from multiple sperm, which it does by using sodium ions to set an electric fence around the cell. Then it releases calcium to, among other things, harden the outer vitelline membrane surrounding the egg and make it impenetrable to other sperm. Once it's protected itself, it needs to rev up its metabolism and start dividing, but it can only do that by releasing all of its zinc ions in a series of strong bursts. Scientists call these sparks. Zinc sparks have been observed in a bunch of other mammals, but until this study, we weren't sure whether it occurred in humans. To determine whether we do it too, scientists bathed human oocytes in a petri dish of fluorescent dye that can only emit light when zinc is present. At the beginning of the experiment, all the zinc was inside the cell, so they saw no signal. but then they injected an RNA construct into the oocytes to emulate fertilization, and it worked. They observed a burst of light from around some eggs, indicating their zinc had been released. To be clear, scientists didn't want to make actual humans in the lab, and they didn't actually fertilize the eggs with human sperm. Instead, they used a synthetic blank RNA construct common in this type of experiment. From the study results, the scientists were able to conclude that human eggs, like those of mice and non-human primates, do exhibit the zinc spark, which is cool. And the zinc signal can be used as an indication of health and embryonic progression after fertilization. This is even cooler. If the technique were to migrate from the lab to the doctor's office, it could have a significant impact on in vitro fertilization, or IVF, because that spark is proof the progression into an embryo has begun. Well, you will probably never see these zinc sparks. They're happening every time a sperm successfully fertilizes an egg. This spark of life can tell researchers and IVF doctors which eggs are good. This is helpful because IVF normally involves artificially bringing together harvested eggs and sperm from potential parents. After they meet, the zygotes are implanted into the woman's womb. Then everyone crosses their fingers, and they often implant more than one to increase the odds of success. Even still, the CDC reports women under 35 have the best chance for IVF success, but that rate is only around 40%. The ability to visualize these newly discovered zinc bursts at the time of fertilization could guide selection of healthy, successfully fertilized eggs for implantation, improving pregnancy success rates, reducing the need for repeat procedures, lowering costs, and easing the long, expensive, and emotionally draining process for potential parents. Go science! Embryonic cells can be used for a ton of amazing things. Learn more about how we can grab stem cells guilt-free in Julian's video here.
Prenatal development. So fewer than half of all fertilized eggs, called zygotes, so all fertilized eggs are called zygotes, survive beyond the first 2 weeks. But for you and me, good fortune prevailed. One cell became 2, then 4, each just like the first, until the cell division had produced some 100 identical cells within the first week. Then the cells began to differentiate, to specialize in structure and function. How identical cells do this, as if one decides I'll become a brain and you become the intestines, is a puzzle that scientists are still trying to solve. About 10 days after conception, the zygote attaches to the mother's uterine wall, beginning approximately 37 weeks of the closest human relationship to exist. The zygote's inner cells become the embryo. So at that point, about 10 days after conception, the zygote becomes an embryo and is attached to the wall. The outer cells become the placenta, the life link that transfers nutrients and oxygen from mother to embryo, and a healthy and well-nourished mother helps form a healthy baby to be. Over the next 6 weeks, the embryo's organs begin to form and function, and the heart begins to beat. By 9 weeks after conception, an embryo looks unmistakably like a human, and it is now a fetus, Latin for offspring or young one. During the 6th month, organs such as the stomach have developed enough to give the fetus a good chance of survival if born prematurely. The placenta transfers nutrients and oxygens from mother to fetus. The placenta screens out a lot of harmful substances, but some slip by. Teratogens are agents like viruses and drugs that can damage an embryo or a fetus. This is one reason that pregnant women are advised not to drink alcoholic beverages. A pregnant woman never drinks alone. The alcohol enters her bloodstream and her fetus, and it depresses activity in both their central nervous systems, in both bodies' central nervous systems. Alcohol use during pregnancy may prime the woman's offspring to like alcohol and may put them at risk for heavy drinking and alcohol use disorders during their teens. In experiments, when pregnant rats drank alcohol, their offspring later displayed a liking for alcohol's taste and odor. Even light drinking or occasional binge drinking can affect the fetal brain. Persistent heavy drinking puts the fetus at risk for fetal alcohol syndrome and other problems like hyperactivity and lower intelligence. About 1 in 800 have visible fetal alcohol syndrome effects and have lifelong physical and mental brain abnormalities as a result. An epigenetic effect is when things like alcohol leave a chemical mark on the DNA that switches genes abnormally on or off, and that can happen during this stage very easily. A competent newborn will demonstrate reflexes like rooting reflexes, seeking out the nipple. Habituation, they decrease their response with repeated stimulation of the same thing. They prefer novelty, And they can smell their own mothers and recognize voices earlier than we think.